Could you imagine a world where genetic diseases become a thing of the past? Where altering DNA is as simple as editing a Word document? Welcome to the exciting frontier of gene editing, a scientific arena teeming with possibilities and potential. It's a world where scientists can rewrite the genetic code of life, like poets crafting verses, to create a better, healthier world. At its core, gene editing is a technique that allows for precise, targeted changes to specific DNA sequences within a genome. Think of it as a microscopic scalpel that can cut out undesirable genetic traits and replace them with desirable ones. With this power, we could potentially eradicate genetic diseases, improve crop yields, and even produce sustainable biofuels. Imagine a world free from the clutches of diseases like cystic fibrosis, Huntington's, and sickle cell anemia. Gene editing has the potential to make this a reality by targeting and altering the faulty genes responsible for these conditions. But the potential of gene editing extends beyond human health. In agriculture, gene editing could help us develop crops that are more resistant to disease and harsh weather conditions, increasing food security globally. The possibility of creating plants that produce higher yields or that are enriched with essential nutrients could revolutionize the way we feed the world. And let's not forget about energy. As we grapple with the effects of climate change, gene editing could be a game changer in creating sustainable biofuels. By tweaking the genes of certain algae, for instance, we could significantly increase their oil production, providing a greener alternative to fossil fuels. From the human body to the fields of agriculture, from the depths of the ocean to the vastness of space, the potential applications of gene editing are as diverse as they are promising. This may sound like science fiction, but with the advent of CRISPR technology, it is becoming a reality. CRISPR, short for clustered regularly interspaced short palindromic repeats, is a gene editing tool that's changing the game. But how does it work? Let's break it down. Imagine a bustling city, your body. The buildings, roads, and parks are your cells, and the blueprints for these structures are your DNA. But what if there's a mistake in the blueprint? A faulty design that causes a building to crumble or a park to wither? That's where CRISPR comes in. Now imagine a team of architects. That's the CRISPR system, equipped with the most advanced tools. Their star player is a protein named Cas9, the molecular equivalent of a master builder. Cas9 can read blueprints, detect mistakes, and even fix them. But how does Cas9 know where to look? That's the job of another crucial part of the CRISPR system, guide RNA. Think of guide RNA as a GPS, guiding Cas9 to the exact location of the mistake in the DNA. Once there, Cas9 makes a cut in the DNA strand, just like an editor marking a typo in a manuscript. But what happens after the cut? The cell's natural repair machinery kicks in. It can either glue the DNA ends back together or insert a new piece of DNA in the cut. And voila, the mistake is corrected, the blueprint updated, and the city, your body, continues to thrive. But remember, CRISPR is still a young technology. It's like a newly minted architect, fresh out of school. It's making waves, yes, but it's also learning, growing, and refining its techniques. And like any technology, it's not without its challenges and controversies, but we'll get to those later. In essence, CRISPR is like a molecular pair of scissors, cutting and pasting DNA with unprecedented precision. And as we continue to unlock its potential, who knows what incredible breakthroughs lie ahead. With such a powerful tool at our disposal, scientists have made some truly groundbreaking discoveries. When we talk about breakthroughs and applications of CRISPR technology, it's like opening a treasure chest of scientific achievements. One of the most high-profile uses of CRISPR is in the treatment of genetic diseases. This was a realm of medicine that was previously thought to be untouchable, but with CRISPR, we've been able to make remarkable strides. For instance, sickle cell disease, a condition that affects millions worldwide, has seen promising developments. This disease, caused by a single mutation in the DNA sequence, has been successfully corrected in human cells using CRISPR. Similarly, in a monumental breakthrough, scientists have been able to apply CRISPR to treat a form of inherited blindness, restoring vision in mice. These advances have opened up a world of opportunities for curing genetic diseases that were once seen as life sentences. But the applications of CRISPR don't stop at human health. They extend to our food supply too. CRISPR is being used to improve crop yields, making them more resistant to pests and adverse weather conditions. By modifying the genetic makeup of crops, we can produce food that is not only more abundant, but also more nutritious. This could be integral in addressing food security issues and malnutrition around the globe. And it doesn't end there. CRISPR also holds promise in the realm of renewable energy. Scientists are using this gene editing tool to modify algae, turning them into efficient biofuel producers. By tweaking the genes of these tiny organisms, we can potentially create a sustainable and environmentally friendly source of fuel. This could revolutionize our approach to energy production and contribute to a cleaner, greener planet. CRISPR is also being explored in the fight against climate change. Scientists are investigating the possibility of using this technology to edit coral genes, making them more resilient to warmer ocean temperatures. This could help preserve our endangered coral reefs, crucial ecosystems that support a vast array of marine life. And in the realm of data storage, believe it or not, CRISPR may have an answer. Scientists are experimenting with DNA as a medium for storing data. With its compact size and long-lasting stability, DNA could be the future of data storage, and CRISPR could be the tool to make it happen. In summary, the breakthroughs and applications of CRISPR technology are as diverse as they are impressive. From curing genetic diseases to improving our food supply, producing biofuels, combating climate change, and even storing data, CRISPR is reshaping our world in ways we could only have dreamed of a few decades ago. The possibilities with CRISPR are endless, and we are just scratching the surface of its potential. While the potential of CRISPR is extraordinary, it also raises some important ethical questions. Should we be playing with the building blocks of life? This question is at the heart of the ongoing debate surrounding gene editing technology. CRISPR's ability to modify DNA sequences with precision brings us face to face with the very essence of life. It's a power that, if misused, could lead to unforeseen consequences. Picture this, a future where gene editing is used to create so-called designer babies, where parents could potentially choose their child's physical traits or intellectual abilities. This raises unsettling questions about eugenics and the potential for a new form of inequality, where genetic enhancements could become a luxury only available to the privileged few. Beyond these societal concerns, there are also worries about the potential for misuse in the form of bioweapons. The ability to manipulate genetic material could, in the wrong hands, lead to the creation of new, potentially devastating forms of biological warfare. These ethical concerns underscore the need for robust, internationally recognized regulations. It's essential that we have clear guidelines to ensure that CRISPR is used responsibly and ethically. This includes transparency in research, strict oversight of clinical trials, and regulations to prevent misuse. Looking to the future, the implications of CRISPR technology are vast. It holds the promise of a new era in medicine, where we could potentially cure previously untreatable genetic diseases. It could revolutionize agriculture, helping us create crops that are more resistant to disease and better able to withstand changing climate conditions. And it could greatly advance our understanding of life itself, opening new doors in the field of biology. But with this power comes responsibility. As we venture further into the realm of genetic engineering, we must ensure that we're guided by ethical considerations, respecting the complexity of life and the potential long-term impacts of our actions. As we move forward with this revolutionary technology, it's crucial that we tread carefully, balancing the immense potential of CRISPR with the ethical considerations it brings to light. The world of gene editing is fascinating and holds immense potential for our future. We've journeyed together through the inner workings of CRISPR, the revolutionary tool that has made it possible to rewrite the very code of life. This technology, with its ability to make precise changes to DNA sequences, has opened up a new era of scientific breakthroughs. We've explored its applications from creating genetically modified organisms to potentially treating genetic diseases. But we've also grappled with the ethical implications, the questions of how and when it's appropriate to alter the building blocks of life. These are complex issues that will continue to evolve as technology advances. But as we stand at this crossroads, it's clear that CRISPR holds the promise of transforming our understanding of life and health. As we continue to unlock the secrets of our DNA, one thing is certain: CRISPR is set to revolutionize our world.